Hello, and thank you for your interest in GrainGenes. In this tutorial, we will provide a way to increase the density of markers around a gene of interest using GrainGenes' comparative mapping tool, CMAP. CMAP can be approached from two ways. The top-down approach begins with the CMAP link on the GrainGenes homepage. From the GrainGenes homepage, click the CMAP link near the top right corner. For nearly 2 decades, CMAP has been the tool used to compare genetic maps in the GrainGenes collection. While CMAP is not a genome browser, it can help to identify regions of shared synteny, identify mapping anomalies, and enrich loci using consensus map, just to name a few endeavors. Select the GrainGenes data source to access the collection of over 4,500 genetic maps from wheat, barley, rye, and relatives. In this example, we want to demonstrate how to enrich the area around the crown rust resistance gene PC53 in oat. CMAP first requires a user to define a reference map. With oat selected as the reference species, click the reference set link to generate an alphabetical drop-down list of all genetic maps available. Individual maps and metadata are grouped into map data records. Typically, they are named like species, year, feature. Oat 2018 PC53 follows this norm. There are 2 maps of the oat linkage group merge 8 from 2 different populations in this study. There are additional display options, and these will be available once the map is drawn. Please note, only the first 2 parents are listed. From the Reference Map Set information table, click View More to open a new pane of links. CMAP then renders the genetic map in an interactive environment. By clicking a locus, PC53 in this example, a new summary page will open with links back into the GrainGenes database. From the locus report page, one can see all of the maps with this locus, links to associated genes, references, and more. In the next section, we will navigate to CMAP from a specific record in the database. A bottom-up approach involves first searching the GrainGenes database for a marker or gene of interest, then by determining if GrainGenes has a map containing that locus. Click the Search and Browse icon near the top left of the GrainGenes homepage. The Search with Google option at the top is a new feature, and will find information in the GrainGenes website as well as a class browser. The class browser provides an organized structure to generate a table of links for the search term. And at the bottom of the page, the current number of records for each data class is reported. The string PC53 appears in the record title for 5 data classes. It appears in genes, a germplasm line, locus, maps, and map data. If you are looking for a mapped gene, look for the locus link in the gene record. PC53 was mapped in 2 populations. To enrich the region around the gene, let's select the map with the locus centrally mapped in the linkage group. By clicking on a map, CMAP will render the image like we saw in the top-down approach. The next 4 slides will illustrate a simple way to enrich the region around PC53. CMAP has many additional features, and users are strongly encouraged to experiment. In this first example, we will select other maps to compare to the reference map. You can select CMAP to draw a new map on the left or the right. Selecting the Add Maps link will populate a new window containing all of the maps with corresponding markers. Numbers in brackets after the map names indicate the number of linkage groups with corresponding markers. Once a map set is selected, like the 2018 OAT consensus map, the window below will populate with the maps containing matches. Merge 8 has 23 corresponding markers, so let's select that. Apparently, the small linkage group with PC53 was reported in an inverted manner when compared to the consensus map. Let's fix that. So scroll down to the options menu below the maps. Click on the flipped button on the reference map. Click Redraw, and the maps now align. Highlighting markers is possible too, so scroll down and type your marker name into the text box. Click Redraw, and now PC53 is highlighted. By exploring the CMAP details of the consensus map, we can discover that Merge 8 has 4,660 loci. So obviously we're missing a lot here. So let's zoom in on the consensus map. So first I want to select the boundaries between 70 and 90 centimorgans on the consensus map. So scrolling down, I'll edit the start and stop positions. And click Redraw. Okay, this is better, but I think we can zoom in a little more. So now let's zoom in between 79 and 81 centimorgans. Right, so we'll just return to that bottom panel, edit those numbers to 79 to 81, click Redraw, and now we have all of the markers in that region that is co-localized on the 2018 consensus map. Now that the 2018 consensus map displays all the markers, just click on a marker name for the feature page. Okay, from here, you can link back to GrainGenes. And from the GrainGenes locus report, look for a probe record, which in GrainGenes is synonymous to a method. And from this probe record, there's a link to the sequence, which contains the actual SNP. Thanks again for watching this tutorial, and don't hesitate to contact us with questions, comments, or suggestions. Cheers!